Hey, everybody. Welcome back to the Doctors In podcast. I'm your host, Nadia Sabeh, president and founder of Dr. Greenhouse. And this is our series, What Plants Crave, where I'm talking to growers and researchers about the plants they grow in greenhouses and indoor farms. My guest today is Dr. John Ertle, who recently earned his PhD in horticulture and crop science at The Ohio State University, studying under Dr. Cherry Kubota. His research focused on the causes and hopefully potential remedies of leaf tip burn and calcium deficiencies in hydroponically grown lettuce, I think specifically with an eye on vertical farms. He now works as a plant physiologist at PP Systems, which specializes in developing instruments to measure photosynthesis and study leaf level gas exchange. I'm really excited for today's episode and to dig into some of the nitty-gritty surrounding the health and quality of plants grown in vertical farms and controlled environments in general. John, it's really great to have you on the What Plants Crave podcast. I've heard you present on this topic many times. I've seen your posters, and I'm really glad that you specifically chose this topic of leaf tip burn for your research. I know a lot of growers deal with leaf tip burn and blossom end rot and other symptoms of calcium deficiencies, and I'm sure they are all going to be sitting at the edge of their seat wanting to learn more about what they can do in their own operations to limit and avoid calcium deficiency. So welcome to the podcast.
Well, thank you so much for having me on. It's really a pleasure. And we go back quite a few years now. So I'm sure you've seen plenty of my tip burn presentations, but it is really great to be here today and share that with you. Thanks for having me on.
Absolutely. Yeah, it's been fun to see the research progress over the years too, to get to your final conclusions. So I want to start with, why did you choose leaf tip burn?
Yeah, so actually that story does go back a little bit before my graduate education, which I think is kind of fun. When I was an undergraduate, a couple of friends of mine I took biology with actually dropped out of school to start an indoor vertical farm in Akron, Ohio. And so I went to the University of Akron, and about 5 minutes down the street, there was a business incubator that the city of Akron started. And as part of their promise, they gave uh, fixed cost utilities for I think, a 5 year lease on space in the building. And this was a converted old tire factory, and they built this building to withstand, I mean, just crazy things. They thought it was actually a bombing target during the World Wars because Akron had at one point manufactured all of the tires in the world. And so there were like 15-foot high ceilings with these thick I beams and concrete floors. And they ended up getting this space and built out about a 10,000 square foot vertical farm there, which I started working at. And we grew a ton of lettuce and basil and microgreens. And one of the first real big problems we encountered was lettuce tip burn, and we were failing crops with this nutrient disorder that we really didn't know much about. And so we kept trying different cultivars of lettuce to see if it would maybe work out. We would find one that was like really resilient against tip burn, and we could primarily grow that and maybe a few other cultivars. But it just turned out to be such a long process to figure that out. And I thought, wow, this would be a really interesting topic to research in graduate school. And as it turned out, I ended up in Cherry Kubota's lab at Ohio State, where I first did a master's on a totally different topic of fruit and vegetable crop grafting. But then I had an opportunity to work on tip burn as my PhD project. And so I jumped at that chance and was able to actually fulfill one of the initial things that got me so interested in this industry.
That is an amazing opportunity and an amazing timing and synergy
Mm-hmm.
of, and also I think just an amazing example of how research can help answer questions and the challenges that are faced by growers, right? By actual
Absolutely.
practitioners of what we do. So that is an amazing connection. And so can you tell us all, what exactly is leaf tip burn?
Yeah.
And how do you even identify it?
Sure. So you can kind of tell leaf tipburn from the appearance. You see this necrotic tissue formation on the margins of leaves. It looks black. It almost looks like you took a lettuce head and turned it upside down on a stove for 5 or 6 seconds and just kind of crisped up the some of those leaves. But what's actually happening is that the plant has to transpire water to these expanding, growing leaf tissues. And as it does that, it's transporting calcium with that water that's transpiring through the crop. And the magnitude of water transport to any given area is going to increase the magnitude of calcium transport. And calcium is used as this linkage component in the cell wall to give it structure and rigidity. But when a deficiency occurs and there's not enough calcium to hold that structure together, it starts to fail and these cells lyse and they break open. And unfortunately, once a cell is completely destroyed in that way, you can't really grow past it. It becomes an injury to the plant that restricts the growth. But mostly we are worried about it because it reduces the marketability of that lettuce. It gives it an appearance that looks kind of diseased to the average consumer or damaged, and they don't want to consume something that has this necrotic black tissue along the leaf margins because just the appearance is not quite there for the consumer to readily purchase that item with that defect. So
It's interesting. I never thought about leaf tip burn and this calcium disorder as being sort of a mechanical breakdown of the cellular structure of a plant. That it's a damage to the plant. But when I think about looking at leaf tip burn, yeah, it does kind of look damaged.
It does. And the calcium kind of gives it resilience against changes in pressure, right? And so when you have more water flowing to a given area, you'll have this pressure change. And what's happening is there's a specialized cell type called a lenticifer, where that cell type is kind of known to explode under this pressure when the calcium is not quite there to support it. And so then you start leaking. Basically these cell structures are being compromised by this lack of calcium, and then when the plant is going through its normal growth habit, it ends up running into an issue where the cells start breaking down, and so then it's not able to really grow past that point.
So is it like osteoporosis of the leaf?
Almost, yeah. So with other nutrients that are mobile, so calcium is considered an immobile nutrient, which means it's transported with that water as it's flowing through the crop. But other nutrients like nitrogen, for example, can be recruited from different areas of the plant where there's plenty of nitrogen to support a low nitrogen tissue. But for calcium, there's no circulation system that can really direct that water flow anywhere. And so the plant doesn't really have a choice in bringing it to a certain area. It just has to go where that water is flowing through the crop.
I'm really glad that you touched on that because that was going to be one of my questions is how calcium is different from other nutrients that plants are taking up right from the ground or transporting through the plant. Like why is calcium so different from the other nutrients?
There are a few other nutrients that do fall in this category of immobile, but we typically don't see as many deficiencies for those nutrients. Usually there's plenty of nutrition to go around, especially in this kind of hydroponics indoor farming world. It's not common to run into those nutrient deficiencies for leafy greens because we've done a really good job of creating nutrient solutions that are nicely balanced and help assist the plant in growing to its maximum capacity. But for calcium, it's not really a nutrient balance in the root zone issue. It's a nutrient flow issue that's controlled kind of by the physics of the environment and what motivates transpiration from the leaf. And so these small inner leaves that are kind of blocked by the surrounding leaves just don't get enough airflow or other, you could prime transpiration in other ways, with direct lighting sources or supplying a heat load to that area. But this isn't something that is really easy to do necessarily in those indoor farm structures. And so we commonly run into that disorder just for calcium is one of the primary issues for indoor grown lettuce.
So you call calcium immobile. And
Mhm.
so that means that it can't move itself from a place of high concentration to low concentration, right? Like it doesn't move through normal sort of osmosis and concentration gradients,
Mhm.
right? And
Yes.
so it has to be moved in a different way. So one of my favorite topics, tell us how is calcium moved through the plant?
Right. So initial uptake, as always, in the root zone, pushing into the xylem, and then as that xylem flows water out to the extremities and the leaves themselves, pushing out towards the tips of the leaves, we see nutrients getting moved throughout each of those individual leaves through that kind of pumping of water up through the whole plant in the xylem. Um, and so that calcium, the ultimate fate goes wherever the water is flowing quickest, and it's relative. So if you have an area with high transpiration and an area with low transpiration, you're more likely to see a deficiency wherever that low transpiration area exists. And as the lettuce grows throughout its normal life cycle, the shoot biomass kind of outpaces the root growth. And so you have to support this bigger and bigger shoot biomass with a more and more limited resource pool as the roots are not able to keep up with that rapid growth. And so you run into that critical deficiency somewhere sort of towards the end of the growing cycle, which is uh, a large issue because you can't really resolve the problem at that stage. Once tipburn happens, there's no salvaging it. The tissue can't go back to healthy. So it's really just a balance of that shoot and root biomass and the growth rate of the plant versus the calcium supply that's available.
Um, so are there certain plants that are most susceptible to leaf tip burn? You've mentioned lettuce, and I think you
Mm-hmm.
mentioned also the inner leaves of the lettuce.
Mm-hmm.
And are the inner leaves of the lettuce, you mentioned airflow. Is it just airflow, or is it because the more mature leaves have more stomata and maybe more pathways for calcium to flow to it than the younger plants haven't quite developed those pathways yet?
Yeah, so that's a good point. So at a certain point, the oldest leaves are going to reach their full sort of expansion, right? And so then they're not actively expanding that tissue like these young leaves are, which are experiencing rapid growth because there's so much more, I guess, source to supply those young leaves with everything they need to grow quickly. But that calcium deficiency comes in once that growth rate increases past the demand, or I'm sorry, increases past the supply of calcium that's available. And so this commonly happens in lettuce, but we also could see it in cabbage. We could see other versions of this, like blossom end rot in tomatoes. And so this calcium issue could pop up other places, but we see it really commonly in indoor farms, and more so than we see it in other growing systems like greenhouses can still struggle with this, and open field growers can still struggle with this. But in indoor farms, it just becomes such a major problem because of the different number of species that can be grown profitably in those indoor farm spaces.
Expand on that a little bit. So you're
Mhm.
saying that the so if we grew the same lettuce plant in a indoor farm or vertical farm,
Mhm.
and that same plant in a greenhouse and the same plant outdoors. Okay, Gi- give me
Sure.
some leeway here because I know that
Yeah.
we have different genetics and different cultivars a lot of times for these different well, we're getting there anyway for vertical farms,
Mhm.
but for these different right growing systems. But you're saying that in a vertical farm, they're more susceptible to leaf tip burn than in a greenhouse and outdoors?
So what we kind of have is an inverse relationship. So as if we think of an open field, greenhouse, in an indoor farm growing the same lettuce, we kind of have a transpirational flux that's highest for the open field, a little bit reduced for the greenhouse, and then majorly reduced for the indoor farm. And essentially, what that is the plant's interaction with the physical environment. So in the open field, uncontrolled temperature, you have a lot of sunlight, you have wind, you have abiotic stresses that are affecting these crops. And then when we move into a greenhouse, by applying a lot of controls, we can increase the growth rate of these crops, shorten that cycle, and have some amount of protection that reduces the transpiration a little bit. And then when we go into an indoor farm, we're highly controlled. We have much slower air movement, much tighter control of temperature, and CO2 can be added in the greenhouse too. And so what we're essentially doing is we're increasing the growth rate as we go from the open field to the indoor farm. And then we're decreasing the transpiration rate. And so that increases our tip burn risk the more controlled we get, essentially. So we're making the plant grow faster by controlling the environment more, and that causes us to run into an issue where transpiration is just reduced enough that we have to now deal with this deficiency more often.
Interesting. So it's about the rate of growth, not necessarily that transpiration is higher or lower. It's just, is transpiration able to keep up with the rate of growth? So there's
Mhm.
some imbalance in our rate of photosynthesis and rate of transpiration.
Yes. So if you have the growth rate as your kind of demand, um, your supply is the root uptake and root pressure and the transpiration rate from the leaves. And we can think of the plant as a big bulk transpiration machine where it's going to be churning out water all the time. But in that area near the meristem, which is centered in that head of lettuce, those small leaves start to initiate and are kind of blocked by the surrounding larger leaves. And so we can have an issue where we're just increasing the growth rate, and that's surpassing our supply of calcium, or we can have some physical kind of barrier that exists when we're not managing the environment properly. And sometimes it's a combination of those two things. Growth rate's very quick, and there's not a lot of air movement, or the environment isn't balanced properly, and we run into that issue.
Okay. So this all makes sense to me and is a great segue into your research.
Mhm.
Because I think some of your research was actually looking at, well, ways to slow the plant down a little bit, right? And so tell us about your research. What environmental
Yeah.
factors were you looking at, and why did you choose those particular factors?
Yeah. So what I wanted to do was first create a condition in which tip burn is certainly going to happen, right? So I used a growth chamber, a couple of walk-in growth chambers. And what I did during the daytime was I added CO2, so about 1,000 ppm of CO2 during the day. I had, a relatively high day temperature. I had relatively high humidity to kind of suppress the transpiration a little bit, and my airspeed was super, super low. I mean, less than zero point 0.1 meter per second
Like
in any direction.
immeasurable.
Just tiny.
Okay.
Like I'm not sure if it was
Like you
error from the
breathe and it
instrument or
changes.
real.
Yeah.
Yeah, I mean, really, I would set up the little anemometer and then I would duck underneath the cart so that it would measure it without my effect. But
Nice.
but really, what we ended up doing was developing this sort of forceful tip burn encounter. And I grew 10 cultivars of lettuce initially, a mixture of ones that were recommended for indoor growing or had some susceptibility to tip burn, and a couple that were reported as very low susceptibility to tip burn. And on the first try, I got every cultivar to have tip burn just horribly. I mean, you wouldn't be able to sell any of them. I think I had like 90%, almost 90% of all of the plants had tip burn in the initial study. So we got it to have tip burn. And then we wanted to work on how can we reduce it from here. So that kind of inducing tip burn environment is what I based all the other experiments off of. So that way, if I saw an effect of trying to reduce tip burn, it would be a little bit more apparent than if I was growing in kind of a normal indoor farming environment.
And to
Um,
kind of just really quickly go back to something earlier you said, which is once you have leaf tip burn, there's no recovering from that. So it's
no
not
coming back.
like you gave these plants tip burn and now you're like, okay, now let's fix these plants. No, when
Mm,
it comes to tip burn, it's all about prevention, right?
correct. Yes. So the next step was a study where I looked at how long it would take for these plants to get tip burn in the indoor farm. And in the indoor farm, you have usually about a 2 week seedling cycle, and then 4 weeks of post transplanting growth in your hydroponic systems. And by the end of that total of 6 weeks, you have this nice 100 gram plus ready to go head of lettuce that looks amazing. Um, and now I've made them all get tip burn and look horrible. And we thought that so lettuce kind of grows as an exponential curve if we think of the growth rate. There's a lot of slow growth, and then the further you go, the more and more lettuce is produced per day, essentially. So by the end of that growing cycle, um, that lettuce is getting real big, harvestable size, ready to go to market. Um, and if you let it go much beyond that, they'll sometimes go to flower, right? Which we want to avoid. So during the last 12 days of this cultivation, I just dimmed the lights. Went from some 17-ish moles a day down in increments of 15%. So I have four treatments, pushing 17 moles a day, 85% of 17 moles, 70% of 17 moles, and 55% of 17 moles just for the last 12 days when the growth rate is at its peak, or is at it, or
Hmm.
increasing per day at its highest. And we know that tip burn happens with about 8 days left in the cultivation cycle. So we pushed it back to about 12 days that we initiated the treatment. And what we saw was that you do get a yield loss. So somewhere, 22 to 26% yield loss with the cultivars I tested by dimming the lights. But for one cultivar, we completely eliminated tip burn. And for the other cultivar, we removed a significant amount of that tip burn to the point where you could almost sell the product as a cut leaf lettuce and just pull off some of that tip burn material, which is a strategy often used by growers to kind of rescue their tip burn crop if they need to. And so I also paired an economic study with this. The cultivar that had tip burn entirely removed actually was less profitable because of the amount of biomass lost. So we're taking that yield loss penalty, but the amount of tip burn we salvaged away from that situation was less economically viable than growing the crop at full strength, selling it as a cut leaf product, and pulling off some of those tip burned leaves. For another cultivar that is very tip burn sensitive, it actually made it more profitable. Only a couple dollars more per meter square of growing area, but you could actually increase your profitability with that cultivar. So this is a good strategy if you have a really sensitive lettuce cultivar that you definitely want to cultivate, even though it has the high likelihood of tip burn happening.
That's really interesting. So not all lettuce plants are created equal when it comes
Definitely not.
to leaf tip burn.
No. The unfortunate thing with that is a lot of the seed companies tend to do their tip burn susceptibility trials in greenhouses, which you
Mm.
do run into less instances of tip burn in a greenhouse than an indoor farm. But broadly, the recommendations are kind of made for indoor farm grown lettuce based on greenhouse research, which may not be true for every seed company, but I know that that is something that I came across quite a few times.
So, okay, what was the lifespan of the lettuce plant? Was it 30 days? Is that what you said? Somewhere in there?
Total, yeah, 28 days post transplant.
20. Okay, okay.
Yeah.
So 28 days and on day 16 then
Yes.
that's when you changed the lighting regime or you lowered
Mhm.
the light intensity to hit that lower DLI. So when you were creating a leaf tip burned plant, were you operating under the same conditions up to that day 16? So
Yes.
you could see where the divergence happened.
The rest of the environment unchanged. I just dimmed the lights. That was the only thing I did to change the growth habit of the plants was just dim the lights. Everything else was still trying to force tip burn to happen, including that low airspeed. And so even with that
Okay.
significantly low airspeed, there was still tip burn happening in some of those treatments.
So when did leaf tip burn emerge after that 16th day? Was it day 17? Was it day 20? Or I'm sure it depended
Mhm.
on the cultivar, like everything, but
Yeah. For most lettuce, we see tip burn emerge somewhere between day 18 and day 22. In this case, for a few of the cultivars, it was a little bit delayed. Instead of day 19, I'm seeing a few pushed to day 21. The average days until tip burn appears kind of creeps up a little bit, but not anything super significant. You still can run into it. So that super sensitive cultivar had actually the same amount of tip burn until I got to the lowest lighting treatment. So until I took out 45% of the photons, um, it had really severe tip burn. I think 30% of the leaves or so or more in that case. But with that lowest treatment, it was down to just a few percent. And that allowed it to still be profitable in that scenario.
so why are people growing that other cultivar?
Yeah, I mean that's a great question. A lot of growers will pick cultivars based on what they think their kind of market demand looks like and what they want their products to look like. So in this case, the super sensitive cultivar had red leaves, really pretty nice leafy cultivar that if I wanted to do a cut leaf or a whole head product, I mean, it was just perfect to rip all these leaves off of and use in those various applications. But it was just so sensitive to tip burn that I thought, why would I ever grow this again? And actually, it was maybe my least favorite to grow. So, sometimes it produced like 90 leaves, this one small cultivar. But
Wow.
um, yeah, but different growers will choose different cultivars based on what they think is best. And I know from experience when we bought a bunch of lettuce cultivars, we were buying packs of like 5,000 seeds and then finding out that that cultivar is super susceptible to tip burn and I can't grow this here profitably. So those seeds went back on the shelf and we got something else. But that is a challenge to run into if you're a grower. And I'm not sure what seeds to buy. Um, Looking at those tip burn recommendations and still finding out that some of them are more susceptible than I was thinking they would be.
So in some ways, your study is looking at ways to allow these growers to produce the pretty plants, right? The crops that they want to sell to market and still have a marketable product to sell
Mhm.
at the end of the cultivation cycle. And it's interesting to me because I mean, there are so many indoor growers, right, who are selling baby greens and teen greens. And I
Mhm.
presume this is a big reason why.
Yeah, I've talked to a number of growers that have switched to that kind of short cycle production. The and I know somebody in Cherry's lab, an undergraduate, Lydia Krage, she did a study about this when I was at Ohio State, and um, the economics of which are really challenging because even though you're shortening your production cycle, you have to have a much higher planting density. You have to pay more in seeds. You have to have more substrate available to grow those seeds out before you transplant, and then by the time you get to the end of that production, you're looking at similar biomass as just growing full heads of lettuce separately. So that issue can be economically challenging to kind of try and balance. And growers are really just looking for ways to kind of get around this tip burn problem because it's difficult to sometimes redesign a farm to have better airflow or to come up with new strategies to help out this crop without, really throwing a lot of time at this problem to try and solve it.
Yeah. Okay. So low DLI had mixed results. It depended on the cultivar. One
Mhm.
cultivar, it was fine to grow less yield because you could make up for it in reduced lost
Re-
product, right?
Reduced lost product and reduced energy cost for using less light. So
Oh, even
that
better.
was
Love that.
yeah. So
Love
that's
that.
that's where
Yes.
that economic benefit comes from. But if you're growing just a slightly sensitive cultivar, you might lose more biomass than tip burn you've averted. So
Then you can
if you
even recover from lower operating costs and I guess not you don't have enough extra marketable product to
Mm,
make up for it. Okay, so
yes.
that's DLI. So we said, okay, so plant, you're going to slow down because we're going to give you less light during your photo period. Okay, so what's the next experiment that you did?
Yeah, so this one is interesting. We kind of went outside of the box with this one. There is a very interesting mechanism to open stomata using blue light. So stomata are typically open one of two ways. One is photosynthesis begins happening. The plant says, "Oh my God, we need CO2. Let's open the floodgates and bring it in here." The other method actually is something that affects phytochrome and cryptochrome inside the leaf, essentially forcing guard cells to swell and opening the stomata. It's not as effective at opening stomata as real photosynthesis is. But if I supply just a little bit of blue light to these plants, I found that the stomates would open and it would increase the transpirational flux over a long period of time if you keep continuously applying that blue light. And so what we wanted to do was see if we could grow normal daytime cycle and then add in just a little bit of blue light, just about 30 micromoles per meter square per second at during the nighttime. So
Is
this
that
is
like
something that,
moonlight?
yeah, a little bit more than moonlight. Yes.
Okay, okay.
I could ask Bruce for his measurement of moonlight, I'm sure, but I think it was tidy. And in this case, it was 30 micromoles is not a lot for the plants. There's the light compensation point is when there's enough light for the plant to use in photosynthesis that it starts positively producing biomass. Um, if it's less than that amount, the plant is respiring and it's actually losing biomass. And so we wanted to make sure we weren't increasing the growth rate of the plant because that contributes to calcium demand. So we did some light response curves and found that if we use 30 micromoles of light, we're not really demanding the plant to grow with that amount of photon flux. The plant is still respiring, but there's a little bit of photons at play that at least keep the stomata open more than they would during the nighttime.
You're kind of propping open their mouths a little bit.
Just a
They're
little
mouth
bit.
breathing at night instead of nose breathing.
That's right. And so the hope was that, a grower could just put in a cheap string, of LED strip lights or something that are just blue in color, and they could turn them on at night and hopefully revert or avoid a lot of the calcium deficiency that could happen in their farm. What we ended up finding was that in this tipburn inducing condition, the airflow was just so low that even though the stomates are wide open, there's not enough physical enticing to flux water through the plant. And so it didn't really do what we hoped it would do. There was no real reduction in tip burn. But once I added in normal indoor farm fans, it quickly took care of any tip burn that would have appeared. So basically, the first experiment was just applying this blue light and a mixture of blue and red light, still at that low intensity. We didn't see any effect from those on tip burn. We did see increase in transpiration and increase in stomatal conductance, but it wasn't enough to really avoid tip burn. And then we did a follow-up study where we had the same treatments, but with or without fans. Those fans moved that vertical air to about 0.4 meters per second, which is the typical kind of recommendation that we give to growers who are trying to grow lettuce tip burn free. And if there was really a strong effect of this blue light, I would have seen a difference between blue light with fans and just fans in nighttime darkness. But no real difference there. So the blue light contributes just a tiny bit, but nothing really significant to push transpiration of the crop during the nighttime. So unfortunately, that one didn't pan out as well as I'd hoped, but it was still an interesting finding to try and apply this mechanism and see if it would provide any benefit to the growers.
So it was airflow really that helped to move transpiration through the plant, to move water through the plant. And you did see a reduction in leaf tip burn when you applied blue light and airflow?
Yeah, so with blue light and airflow or nighttime darkness and airflow, I got similar results. So it
So
seemed
you didn't
that
even so you didn't even have to prop the stomata open to reduce leaf tip burn. Is there transpiration happening if the stomata aren't opening, or the stomata opening just as a response to air movement?
No, the stomata are opening, and they're opening in response to the blue light. So it's really cool. I can just put a leaf cuvette with a gas exchange analyzer on the leaf and apply that small amount of blue light, and you can see the stomates open and continue being open. So sometimes like 25 to 50% increase in stomatal conductance with that blue light, which is amazing actually to try and apply that, and it works. And then on the back end, it didn't really assist with the transpiration as much as I was hoping to, because what we were doing was we were increasing whole plant transpiration. The whole plant is getting this blue light signal, it's opening the stomata, but because water only flows to where there's really transpirational, there's a flux happening, where transpiration is actually happening, those inner leaves are still just so blocked up by the structure of the lettuce itself that we're not getting a lot of transpiration in that area. So what we were doing is we were opening all the pores, all the stomates on the plant, but it's not really driving calcium to the area where we need it, and so it didn't quite work. But other studies have done kind of similar mechanisms where they put like perforated air tubes over the crop, and they only blow air at the center of the head to increase the transpiration rate. And that works remarkably well. But in this case, since we're applying the stimulus to the whole plant, it's really not pushing that transpirational flux to that area we need it. It's making the whole plant transpire more.
So you just need to stick one little blue diode into the center of the plant.
Yeah, I mean, it would be really cool to have a precision lighting strategy that really works that well, it'd be awesome. But unfortunately, that didn't pan out here in my
That'd be
broad lighting
pretty,
strategy.
yeah. So, but you said also in the dark without applying the blue light, did you say that airflow also helped to reduce leaf tip burn?
Yeah, and so I was able to confirm what a lot of other studies have said is that airflow over this crop, especially when the air is moving vertically downward at the crop, it's breaking up that boundary layer near the meristem and the young leaves and facilitating good transpiration there. That's pulling calcium to the area and averting any of the nutrient deficiencies that we would normally see. And horizontal airflow experiments haven't been as highly successful as these vertical airflow experiments. So what I was hoping to see was maybe a big difference between just blue light and blue light in fans at night or darkness at night. And I didn't really see that effect. Since I'm growing in this really tipper and inducive condition, I was hoping that, maybe we would see that effect in maybe not in the experimental version where I have this low airflow state, but in a higher airflow state, we would hopefully see that big blue light response contributing to tip burn avoidance, and maybe for really sensitive cultivars. But unfortunately, that didn't pan out as I had hoped, and there wasn't any real significant difference between nighttime darkness with fans and blue light at night with fans. It's not super practical for growers to apply that strategy, even if it works.
Mm,
But unfortunately, we didn't get to cross
yeah.
that bridge.
Under the when you were applying the airflow, were you only using it at night, or were you also applying a higher airflow during the daytime hours during the photo period?
I did apply it during the day as well, and I'd be curious to do one where I just apply it at night. We were thinking of doing that experiment, but there aren't really any growers that can just insert airflow only during the nighttime and
That's true.
remove it during the day. Um, so that would be an interesting study. So I'm thinking I may end up doing another tip burn study in the future. So
Just do
we'll
it on
see.
your own, yeah. So, was there another experiment that you did?
So besides that, I did another cultivar trial experiment just to see what plants were susceptible versus less susceptible to tip burn. So I ended up pulling another 20 cultivars or so. I think I tested a total of, somewhere around 30 total cultivars on the market. And this time I asked for ones that were specifically recommended for indoor growers. And
Mm.
I just looked across this scale of about 20 different cultivars, and I found five that in this tip burn inducing condition have less than 10% tip burn rate. So less than 10% of the leaves have tip burn. But for 15 of those cultivars that are commonly recommended to growers, there was more tip
Interesting.
burn, somewhere sometimes up to like, 50 or more percent, which is
Wow.
great.
Scandalous.
So yeah. Well, I mean, I'm growing them in the worst condition possible.
True,
So
true.
the average grower is hopefully going to have better results than I did. But if anything is developing less than 10% tip burn in my condition, I would be pretty confident in saying that it will do very nicely in an indoor farm setting.
But see, I love that because that's what we need, right? Is we need seed catalogs that
Mm-hmm.
represent, the true response that we're going to get from the plants in this very unique and new type of environment that we're trying to grow in. So I
Right.
hope that you had some positive feedback from those seed producers, both that succeeded, that passed your test and didn't pass your test. Like, thank you for doing, this independent research, so we can, you know take it from there, type of thing.
I actually had a lot of nice support from the
Good.
seed companies that I talked with. A lot of folks are now trying to do more breeding for CEA specifically. And so anytime we're involved in that process, feedback, telling them how our experience with these plants and their cultivars are, and giving them information to feed back into their programs, I mean, is really great to kind of catch up our industry breeding wise to some of the other industries that exist. And I'm really excited about what the future of that is going to look like as we keep going in that direction.
Yeah, and to me, I mean, your research really comes full circle because starting as a grower, having this challenge, seeing this issue, and then taking it to the research, right? And saying, okay, what can we do to make this better? And then being able to communicate back to industry partners to say, this is what works, this is what didn't work, right? To me, it's just perfect. The perfect circular
Mm.
research industry type of study.
Full
Yeah.
circle experiment. It was really, really awesome. And I'm still kind of in disbelief that I got to work on the very problem that really made me
Oh,
interested
yeah.
in this field in the first place. So that
Yeah.
was just cool all around to be able to do that.
Okay, so I have to ask this question because I
Mhm.
have been wanting to ask this question from like the very beginning of the conversation, and I cannot be the only person just dying to ask this.
Mhm.
Can you just add more calcium to the nutrient mix?
This
Right?
is
I mean,
yeah, this is such a great question that I get so often. But yes, so what we found was that when we do add calcium to more calcium to the nutrient solution, it doesn't really aid in that directional xylem uptake flow pathway. So we can keep adding more calcium, but eventually we'll run into some kind of salt stress. Actually,
hmm,
usually before we run into some benefit for the calcium uptake. So calcium and actually magnesium share one uptake pathway, and magnesium has a second uptake pathway where calcium kind of has to compete a little bit more. And so calcium being taken up through the xylem is, for the most part, pretty straightforward, except when the plant starts outpacing the calcium uptake. And it turns out that putting more calcium in the root zone isn't really increasing the rate of uptake, which is unfortunate because that would be the fastest, best, easiest solution. A handful of experiments have very thoroughly shown that it just isn't going to give us the benefit we hope it would.
I mean, I think earlier you said something that might attribute to that, which is, you said in hydroponics, especially, right?
Mhm.
The leaf growth can outpace the root growth. And so I just imagine all these, little calcium particles, flowing by the roots.
Right.
And there's only so many roots, right? And so many places on the roots where they can actually take that calcium up. So you become limited, I would imagine, in some ways by the roots themselves.
Yeah. So Yeah, basically the roots have great uptake of just about every other nutrient. And in this case, there's plenty of calcium in that root zone. So actually, even inducing a calcium deficiency is hard to do for lettuce unless the environment sort of restricts that transpirational flux. So when we do have it in this really tightly controlled, fast-paced growing environment, we do run into that problem. But sometimes in the greenhouse, for example, maybe I'm growing in the winter, and I have supplemental lighting, but it's not, I'm not really pushing these crops to the fastest pace I could. I might not run into tip burn at all because the growing processes takes a bit longer and the growth is a bit slower. So you can sort of avert that deficiency entirely sometimes. But in the indoor farm, we're very strict about that, 6 weeks seed to sale kind of process that, we just keep running into the same issue unless you lower the growth rate, which case you can lower your profitability of your farm. And that could be very damaging for an indoor farm with thin margins versus
Mm.
other growers that might have a little bit more room to play with those dates.
So, low evapotranspiration rates can induce leaf tip burn, right? Because
Mhm.
we're, not pulling calcium up, right? Like it's just a slow water movement process. And then you can
Yeah.
have high evapotranspiration rates, which can also, right, affect leaf tip burn and leave some of the calcium behind. Like the water can move so quickly that the calcium can't keep up with it. Is that true?
Well, it's more the direction of the water flow, I think, than
Okay.
the amount of water taken up, right? So, for example, like iceberg lettuce, kind of has a habit of kind of enclosing over itself, kind of like cabbage does, right? The leaves just envelop each other. And sometimes you get really, really bad tip burn in iceberg lettuce because, of course, you can't really transpire from the meristem when it's blocked and protected from any physical environmental parameters that are going to cause transpirational flux. But then there are other cultivars with a very open growing habit that get tip burn just as badly. And some of that is genetics. And we're starting to see more research come out. There's a few good papers in the last few years about, certain genes that are linked to cell proliferation or cell expansion or calcium pumps and ion pumps. But there's kind of a genetic component and there's an environmental component. And even though with this hydroponics, we always have water and nutrients available, this problem just seems to persist in a lot of lettuce cultivars in different environments.
I've heard calcium described as a heavy molecule. And that's why it kind of gets left behind. And I've seen, especially in cannabis facilities where they're sitting right next to a fan, and those plants have leaf tip burn worse
Mm.
than their neighbors that aren't sitting right under the fan. So I think, well, that's
Oh, I
a
see what you
that
mean.
yeah, the transpiration is too high, right? The water is moving so fast and that the calcium just can't keep up. And a lot of times it's at the top of the plant, right? Again, those
Yeah.
younger leaves, like you were describing with lettuce. So to me, it seems like there's, too low is bad, too high is bad, and that's got to be a really high transpiration rate. I'm curious with lettuce, you measure transpiration rates, right?
Yeah,
In a way that
absolutely.
you could. Yeah.
Yeah.
So was there a sweet spot? Did you find that didn't result in leaf tip burn or did result in leaf tip
Hmm.
burn?
Yeah, so that's interesting too. With lettuce, at least, and I think this is true of many other plants, but in this case, I'll just restrict it to lettuce. Each newly initiated leaf has less calcium than the previous leaf, essentially. So as I keep going, if I test every single one, leaf two, leaf three, they're all going to have just slightly less calcium than the leaf prior to it. And in this cannabis example, I'm wondering if maybe that high fan speed is actually causing stress on that leaf where the stomata are restricting themselves, and then there's not transport of water going to that area, and
Uh-huh.
there's not calcium to being deposited, or there's so much air movement and there is transpiration happening, and that water is not reaching all the way out to the margins of the leaves because it's being transpired before it can fully get out to the expansion points. So that could be the case too.
Yeah, nice.
Yeah, but I'm trying to remember this
The
second
original,
half of this
the
question.
second
Yes,
half of the question was there a sweet spot with lettuce in terms of
yes,
transpiration rate?
yeah. So that was something I was really, really hoping to find. Actually, I spent a lot
Mm.
of time doing leaf gas exchange data collection during the day, during the night, during where my sleep schedule was terrible. I was trying really hard to find the flux point at which we know we're going to avert this disorder, and it turns out it's not that simple. It's actually so different for each cultivar, completely different. And even we've kind of historically thought tipburn was associated with a few different things, like maybe leaf color, like green lettuce was more susceptible than red, or morphology, like a romaine lettuce with that heading type that closes those inner leaves is going to be more susceptible than an open butterhead or a leafy type.
Hmm.
Um, and turns out neither of those are the case. Red and green lettuce, no significant effect. Morphology, no significant effect. But the actual transpiration rate of each of these plants was so different and had no real correlation to the amount, the calcium, the amount of calcium in those tissues and the amount of tip burn that they had was cultivar specific, which is
Yeah.
makes it way more complicated. you know,
Confounded plants.
so many. Yes,
They all gotta be
exactly.
their own.
Yes,
That's
very
funny.
individualistic, these lettuces.
Right,
So,
right. Well, and
yes.
also, like you said, I mean, if each leaf has different amounts of calcium, they have different amount, number of stomata,
Mhm.
then even the rate from, the outer leaf, the transpiration rate from the outer leaf to the inner leaf must also be changing, I would expect. So even if you're getting a whole plant transpiration rate, but the issue's in the middle of the plant, like that's what you really want to know, right? Like what the
Yeah.
transpiration rate should be in the middle of that plant.
Well, what was somewhat challenging was that if I measure the transpiration rate directly, that can tell me a lot about how much transpiration I should expect from that leaf tissue, but that doesn't necessarily factor in the expansion rate of that leaf and how much calcium might be demanded given the expansion rate. So, for example, like at the very beginning, leaves do grow more slowly, but as time goes on, that leaf expansion rate just keeps increasing, right? And so it's much easier to run into a deficiency, even though the transpiration rate of the youngest leaves and the oldest leaves are pretty similar on
Hmm.
the same area basis, but one leaf has much more surface area, one leaf has much less surface area, and one leaf isn't really expanding or expanding quickly, and the other leaf is expanding very quickly. And so that kind of confounds the issue. And the other side of it is the physical environment interaction with that plant. So, what is the boundary layer thickness over a given area, over a given leaf area? Um, or what's the boundary layer thickness over just the meristematic area versus the edge of the head of lettuce, and you can get very different answers for that kind of calculation.
I mean, this sounds like this is a super labor intensive and impractical question, but
Mm-hmm.
what if we just chopped off the outer leaves on day 16 and
You know,
focus the energy to the middle of the plant?
it would be interesting. Like, what if we did like a cut and come again harvest? Where you
Mm-hmm.
you take off these outer leaves and then you push the plant to grow some more. Um, and I haven't really seen anybody do that if I think about it. We do it for a lot of other herbs, but we haven't done it really for lettuce. That could be interesting to see, although you might run into issues with the plant wanting to go to flower or things like that happening the longer you keep it in that condition.
Oh,
But you might see
yeah.
something interesting.
Yeah. Eventually we'll have the robots to do that chopping of the outer leaves without it
That's
being.
true. That's true.
Seems very labor intensive.
Yeah.
Do you have any thoughts about the stomata themselves, in terms of their size or concentration? Do you think that having fewer larger stomata would induce more leaf tip burn versus, say, more smaller stomata or vice versa?
I think that's a great question. And honestly, it's hard to know exactly what the kind of stomatal layout and aperture size are for such a wide range of cultivars. That'd be a really, really interesting study to do, and just see what that looks like. But I don't have any, I guess, hot take on whether larger, fewer stomata or more smaller stomata would be better, as long as the amount of stomata is appropriate for the amount of transpiration that plant needs to have to
Mm,
avert the deficiency. And that number of stomata might be completely different for the oldest leaf versus the youngest leaf. And so even if we bred for something like a kind of a fixed stomatal aperture and density, that might be perfect for certain range of leaves, but then restrictive for leaves towards the end of the cropping cycle potentially.
hmm
And so you might even run into things like, well, if I have way more transpiration than I need early on in the crop, maybe I have, a water use efficiency issue. And then at the end of the crop, I have, plenty of movement to reduce or avoid tip burn, but you might take, a penalty somewhere else to accept that trade-off. But that would be cool to see that thread open up a little bit more.
Yeah, I've read some articles that you can induce drought tolerance in some crops, some horticultural crops like tomatoes, by keeping water from them early in their growth
Hmm.
stage, and during the seedling stage. And then as they mature, they use less water. They need less water to function and still produce. And one of the theories that some of those articles or some of that research has proposed is that those plants tend to have more smaller stomata than the
Hmm.
ones that are flush with water from the beginning of their life. So it just kind of makes me think like,
Yeah.
if you have more of these sort of valves or openings, right, then you have more control in a way of how you're using water, where you're putting nutrients to within your leaf, that maybe
Yeah.
it helps the plant self-regulate better to have more regulating mechanisms in that leaf.
That would be really interesting. And you might be right. Maybe we have having a inducing kind of a higher density of stomata would allow it to have finer mechanistic control over just that transpiration where it needs it.
Mm-hmm.
And that would be really cool to see if that application works really well for this process. And it might
Hopefully
be that,
Cherry's listening. Cherry,
yeah,
are you
exactly.
listening? That's the next research project.
Yes, Cherry, we need you to breed lettuce now and fix this problem. Yes, this would be perfect.
I cut you off. What were you going to say? Do you remember?
No, just that it would be. This is a cool idea, and I would love to see if we can avoid tip burn in this very cool fashion.
Yeah.
Uh, driving the small density.
Maybe hydroponics isn't the way. No, I didn't
We
say
will
that.
find
Not
out.
a Dr. Greenhouse did
Oh,
not say that. No. All
yes,
right.
we
Well,
can't alienate all of the controlled environment folks.
yeah, exactly, right. So, John, I really appreciate the time. I mean, spending this much
Mhm.
time talking about one very specific topic on plant health and quality. You know, Tell us a little bit about what you're doing now and how it relates to the research that you did as a grad student.
Yeah, so now I work at PP Systems. So we manufacture, develop, and sell gas exchange equipment to measure leaf level gas exchange, as well as a number of other applications. So soil respiration and even insect respiration we do a little bit of. So just a wide variety of gas analyzer technology applications. And they brought me in as the plant physiologist to start developing the capacity to do in-house research. The company's been around for 40 years this year, and I am tasked with kind of building this lab here and starting to build a little research program now that industry research has become more and more popular. I think I'm in a good position to kind of do some stuff in-house, and then some collaborative things with academia or industry parties. And so lately I've been working on a few things. One, I'm doing a far red study. So we've seen lots of experiments with far red in recent history and kind of their equivalency to PAR photons when we do far red substitution. So we replace some of the PAR spectrum with far red, and we look at the growth of the plant, and we've kind of come to the conclusion that far red can be used photosynthetically, and we actually get very similar growth rates as with just strictly PAR photons. And so I'm kind of measuring that at a leaf level, which has a little bit more variation. And I'm trying to see some trends in different species and crop types to see exactly how that light is interacting with the photosynthetic rate at the level of the leaf, which is really interesting. And then we've also just acquired some more space to grow out some plants and do a little bit more controlled environment research. So I'm the proud owner of a secondhand shipping container that is now outfitted as a vertical farm. And so I am starting to get set up in there, and we'll hopefully have plants in there in the next month or so to do some more research in there. And I'll do an initial grow out to make sure everything works. But then I'm thinking of doing some leafy green studies in there initially to look at photosynthesis under different lighting conditions and changing the environment a bit. And so that there's more that will come out about that. But those are kind of my recent projects that I've been engaged with here.
Something I want to know
Mm-hmm.
is, we see all these curves, these photosynthesis curves looking at, light and CO2 and temperature, sort of this like three-legged stool that, all work together towards photosynthesis. I want to know if temperature, I want to know where VPD fits into the equation. Because
Mm,
I am suspicious that it's not just temperature. For sure, temperature. We know higher temperature, higher metabolic rates. I mean, that we get temperature, but I want to know if we adjust the humidity, and we're trying to track, we say 1 kilopascal for a VPD, or we say, zero point 0.8 for a lettuce and 1 for a tomato and 1.2 for cannabis, pick your
yeah.
plant, right? We're always kind of within this 0.8 to 1.2. But how does humidity play into the photosynthetic rate? And I feel like probably that has not been studied so much because it's really hard to regulate
Mm,
humidity in a growth chamber, that it's just like, we can't do that, so we're just not going to
yeah.
study that. If you guys are able to set up an experiment to look at that, please let me know, because I would love to collaborate with you on that, or just know what
Okay.
the results are, honestly. Um, yeah.
We could definitely talk about that. I think I might have some interesting things for you. But
Awesome.
it is interesting you bring up VPD, and in controlled environments, everybody seems to kind of use, like what you said, like kind of a fixed recommendation for a certain crop type. And there's not really a lot of dynamic control of VPD for any real given purpose. And
Yeah.
I guess from maybe this question on your end is, in the controlled environment sort of segment, are there reasons that people should vary that VPD throughout the diurnal cycle? Not
Mm.
just day to night, but actually during the day, is there any reason to change between, kind of different VPD throughout that normal cycle?
I mean, maybe. I mean, if we look at day to night, we know that there are some plants, right, like strawberries, that's how they, having that really high humidity allows the plants to translocate, right, a lot of those goodies, those good nutrients into the berry. And so, but
Mhm.
we don't necessarily want that every night. Right. I mean,
Right.
it's so amazing how much we're really trying to simulate the central coast of California and what
Yes.
we experience weather-wise here.
Yes.
But it's scary, I think, for growers to have a almost 100% humidity or zero VPD, right? You
Right.
say, well, do it for strawberries for a couple of days, and it seems to not have a detrimental effect on the berries quality. In fact, it seems to be a benefit. But if I
Mhm.
told that to a tomato grower, which I don't even know if there's been research that on, say, tomatoes, that seems a little scary. If I told that to a
Mhm.
cannabis grower, and I have told a couple of cannabis,
Yeah.
strawberries do this. Oh my God. It's just like their eyes look at me like, are you crazy? We're going
Yeah.
to have botrytis and powdery mildew and all these issues if we
Yeah.
have a zero VPD. But it's like you look at strawberries and what they're doing. Is there
Mhm.
something special about strawberries that does that? Or is there just, we just stumbled upon that? And now it's,
Yeah.
we haven't looked at it in other crops that there could be potential benefit there. Um, and VPD is used a lot as one of those sort of stress inducers, I would say,
Mm.
right? Like, a lower VPD is more of a vegetative sort of state, which is what
Mm.
we want with the lettuce crop.
Mhm.
It's what we want with a vegetative cannabis crop. It's what we want at the seedling stage, right? It's what we want for even grafting. It's a really high humidity,
Right.
right? We want these low VPDs, and because we don't want to stress the plant, right? We want the plant to be
Right.
comfortable and feel safe in its environment, and VPD is one of those things that wraps a blanket around a plant,
Mhm.
right? And then when we want to stress that plant and induce the flowers, right? Induce fruit production, induce whatever stress response we want, we rip that blanket off, right? And say, okay, plant, you're going to die. And that's when we might have, this elevated VPD. But there's two ways we can achieve that, right? With a high temperature or a low
Mhm.
humidity. And so it's like there's a point which you can ride sort of the curve together, but I think there's also a separation point where you could have too high of a temperature, and
Yeah.
you might get more heat stress response than the stress flower, produce me flower response. And the lower humidity, now all of a sudden, powdery mildew becomes a big issue, and it's not even easy to
Right.
achieve. So yeah, I mean, we know VPD is important. The Japanese researchers in the 90s were doing studies on that.
Mhm.
And there are some guidelines for some plants, but there just hasn't been a lot of fundamental research on a
Yes.
lot of the horticultural crops when it comes to VPD. And as an HVAC person, if we can find a way to control VPD that uses less energy,
Mhm.
right, creates the comfy or the stressful response that we want, and still induce the crop responses that are
Mhm.
desired, I'm all ears.
Yeah, I think you're onto something there, and I there aren't really a lot of good structured kind of methodologies for why you would adjust your VPD in a given condition and what benefit you might see from that. But for example, one of Cherry's studies actually was about strawberry tipburn in the greenhouses in Arizona. And so they applied this super high VP or super low VPD, so pretty close to 100% humidity during the nighttime for just a couple hours, like 2 to 3 times a week. And that averted all of this leaf tip burn that was happening in strawberries and the calyx tip burn, right? Which happened, the calyx are the little leaves by
Yeah.
the berry itself. And people weren't buying this strawberry with like tip burned calyx leaves because they're like, oh, it's diseased, it's unhealthy. We don't want to buy that. It doesn't look good. It's like, no, it's just, a calcium deficiency on some,
It's
a
just the
little
window
greenery you're not going to eat
dressing.
anyway. Yeah, it's
Yeah.
like it's not, you're not eating it. Um, but like that same strategy, there was a couple of studies in the 90s that looked at that for lettuce, and they did at night, they would take this little plastic hat kind of thing, and they would just put it over the meristem of the lettuce at the start of nighttime, and it had an artificial sort of increase in relative humidity, decrease in VPD under that little dome over the meristem. And that actually increased the pressure that was happening in those small leaves. And they started to force water out towards the hydrothode, which is the little opening at the very end of the leaf that you usually can't see. And so the leaves would actually push this water out to, because of the pressure and there's nowhere for the water to go, it would just stay in a liquid form and literally force out through the edge of the leaf. And by doing so, it actually dropped copious amounts of calcium. in this small leaf meristematic area. And it's really effective for avoiding tip burn. But for a large indoor grower, it's kind of impractical to go around and put a little hat on your hundreds or thousands of lettuce plants every night, even if
But
you just do it for the
imagine,
last week.
but imagine the social media photo campaign if all your little
Oh,
lettuce plants were wearing hats. I mean, that to
yes.
me seems like marketing gold.
It would be. And you're certainly right. Um, but if you're a grower and you want to recreate this strategy just using your the humidification you have in the farm, well, you're going to have to increase that humidification to like 100% for at least your full nighttime and probably most days of the week, if not all days of the week, to actually push that and avoid tip burn. So it could be a really effective strategy if we come up with a better way to manage that sort of water load and avoid the disease incidents that would likely come along with
Hmm.
8 hours or so a night of 100% humidity.
Yeah, but can you imagine telling a lettuce grower to, no, we want you to
Yeah.
target 100% relative humidity. They would look at you like you're
Oh,
crazy.
it might not even be possible.
Well, that's true. And I think there's
Yeah.
also a difference between like the plant creating its own little saturated dome
Right.
versus, spraying water into and having all these free droplets that can basically land anywhere, not just in the center of the plant, but on the racks and on the ground and on, everything
Yeah.
else potentially could become contaminated. I want to ask you though, because you said something that reminded me of a question that I had, which is so that water was forming around the edges. Is that guttation?
Yes.
Okay.
Yeah.
So you get that guttation through what'd you call them? Hydrophone? What'd you
The hydrothode. It's
hydrothode? That is a
yeah.
really
Is
cool
it a fun
term.
word? Yeah.
Yeah. Yeah, that
It's
is
like
the word of the week.
yes, THODE, thode.
Yeah. Like
Is
a
a I've
cathode,
I've
right?
never
Yeah.
heard that ending on another word, so
It's
it's
awesome.
amazing.
Okay, so we have the hydrothodes, and so the water's pushing out of it. And then you said the calcium is in the water and then it distributes.
Mhm.
So is it actually like the water drips down and is distributing calcium to the
It's
surface
actually
of the
that
leaf?
the xylem pressure is high and channeling water through the leaf, and naturally it goes out inside the leaf itself, and so it's being
Mm.
osmotic. There's some symplastic and apoplastic sort of transport happening, but it's almost like rushed because there's so much water and so much calcium backing up that it's just forcing it a little bit more strongly to the through the full leaf, and so the leaf actually swells a little bit. So you can measure the thickness of the leaf, and you can see a swelling under the dome, and essentially as that
Mm.
water is saturating the inside of the leaf, and so it just kind of
Interesting.
deposits this calcium everywhere. Although you can use a foliar calcium spray, so calcium chloride, to
Mm.
achieve the same effects by spraying the leaves. Unfortunately, that kind of broad application is really challenging in indoor farms because you're also spraying, salty water near all of your electrical
Yeah.
equipment and your structures and metal and things that degrade. So it's avoided in most of the indoor scenarios.
For sure. I'm glad you brought up the foliar calcium sprays
Mhm.
because I've talked to tomato and pepper growers who literally take Tums, crush it up,
Oh my
and
gosh,
then like basically pulverize it into a powder, put it in
you're
a
kidding.
spray bottle, with water, shake it up, and then spray literally their tomatoes
Come on.
or their
There has to
peppers
be
with Tums.
there's got to be a cheaper way of getting calcium chloride than crushing up your Tums. Come on.
What flavor
Although,
do you think the tomatoes like, by the way? I don't
I'm not sure. Like, are we talking about like the chewy Tums or like the chalky ones?
I
I
think
think
the
I think the
chalky
chalky
ones.
ones.
Yeah,
Yeah.
yeah.
Yeah. I'm not sure if any of those are really flavors. They're more
More
like
chemicals.
impersonators of flavors, like this one's raspberry. And you're like, I'm not sure
Yeah.
that we've had the same raspberry,
The
Yeah.
tomato's like, get that orange away from me.
Oh God, yeah. Well, maybe you can change, the color of your fruit a little bit by spraying all of this artificial
There
whatever
you
coloration. Something like that.
Okay, but there are official foliar calcium sprays. We don't have to go to
Yes.
the pharmacy and buy some Tums and crush it into a water
Right.
bottle.
You don't have to do that. You definitely can get calcium chloride somewhere else. Really common, really easy
You
to
know, some
source.
farmer was walking around, had like, a pocket full of these because he had, like
Yeah, he looks at the
I
label
don't
on
know.
the back
Yeah.
and thought, "Oh my God, this
Oh,
is it. I've been
calcium.
looking
There you
for
go.
it." Yeah.
Exactly. Well, all right, John, I'm gonna wrap up with my final
Okay.
question. I'm actually kind of curious what your answer is gonna be.
Mhm.
Okay, so what do plants crave?
CO2.
Ladies and gentlemen, he did not say calcium.
I know it's crazy. We crave that too, but
All
I think
right.
CO2 is
Why
a good answer.
CO2? Why is that your choice of all the
Um,
things?
well, they've been fixing CO2 for a long time and evolved this great pathway to do so. And I think that we have a lot to owe plants and their
Mm.
ability to design such a nice little system that provides us with the majority of our calories and the majority of the calories for everything else that's alive and walking around. So uh, it's
Thank
pretty
goodness for
cool of them.
plants.
Yeah.
Yeah. Yeah. All right. Well, that's the end of my official questions, but now
Okay.
I have a few rapid fire questions. So these are
Awesome.
meant to be quick responses. And if you want to elaborate, you can, but they're meant to be quick. All right. Are you
All right.
ready?
Yes.
Okay. Are plants introverts or extroverts?
Extroverts.
What is one piece of advice you'd give to lettuce growers wanting to prevent leaf tip burn?
Have good airflow, vertical downward airflow between zero point 0.3
Specifically,
and zero point 0.5 meters per second, somewhere in there. Yeah.
you heard it here, everybody. What have you learned from plants?
I have learned that they are maybe the most fascinating group of biological things that I could have possibly studied. Um, so I've learned passion from plants.
Nice. I like that. Okay, if you were a lettuce plant growing in a vertical farm,
Mm.
what would you and the other lettuce plants be talking about?
We'd probably talk about the recent outbreak of Pythium a few racks over and how Bob and his buddies look like they have an algae problem down the way. And that other rack looks like it's suffocating without enough airflow. And at least we got plenty of nutrients and light, you know,
I
gossip.
love, yeah, gossip, exactly. And you guys are perfect, I'm sure.
Yeah,
Right? Yeah,
obviously.
your group, yeah, your friends are
Yeah,
perfect.
exactly.
Well, John, thank you so much for interviewing me and or not inter you didn't interview me. Thanks for talking to me about
Maybe
your
one day
Absolutely.
we'll
Anytime if
switch
you ever
chairs.
want to.
Yeah.
Yeah, exactly. I learned a lot. I know that my listeners are gonna just love this episode. So be prepared for some follow-up, I'm sure. And yeah, good luck with your setting up the new lab and
Thank you.
the job that you have right now. I'm excited that you stayed in the field and are expanding on what you've already been doing. Thank you.
Thank you so much for having me. It's great to talk to you. We don't do it often enough, and this is a great opportunity. Thank you to all of the listeners that tune in, and um, I look forward to talking to you soon, Nadia. We'll have a lot to talk about, I think, maybe in the next couple of weeks about humidity and some things there. So that'll
That'd
be
be
fun.
great. Cool.
All right.
All right. Thanks, John. Have a great rest of your day.
Thank you. I hope you have a good one too.
Okay.
Sweet.
All right. That's it. That was
Nice.
awesome.
Thank you. This is actually a lot of fun.
Good.